All right, so, well, thank you very much for inviting me to be here. And I think my only question for Adrian would be, I don't quite understand what was dry about that material, but it's incredibly exciting stuff. All right, so what I want to talk to you today is really, and I want to thank him for getting through all the basics. So I did want to talk a little bit about the future, what FDA's experience has been to date in dealing with some of these new technologies and sort of how we envision moving forward on this, with the understanding, of course, that these are, discussions about policy are preliminary at this point in many cases, and of course, my opinions don't necessarily reflect those of FDA. So, as we've heard many, many times, we've seen a real evolution in technology. And what I want to point out here is just not that we've had an evolution of IVDs and the technology, but also in how we regulate. So, so if we start at the beginning, the classic, what I would call IVD classic, back at the time when the medical device amendments were passed in 1976, and FDA began regulating IVDs, or the tests tended to be more simple. What was cleared and used by labs were often things like test tubes and microscope slides and things like that, not as many kits. And certainly, as was mentioned, HER2 was sort of the big shift in consciousness. We really got the idea of companion diagnostics, even though it wasn't called a companion diagnostic at the time. We started thinking in those terms and eventually moved out, had this guidance come out on companion diagnostics. And certainly that idea was kicking around for a long time and was being utilized within the agency, but the guidance finally became final this last summer. And then we've had a number of new technologies that have been more complicated, but that we have still regulated. We have still regulated and cleared and improved many examples of these. Microarrays, IVD MIAs, so where you're taking a bunch of results and using some sort of algorithm to get to a single result. Multiplex tests, array CGH, those are some examples. They're still, even though they're much more complicated than these single analyte assays, they are still relatively well defined in terms of the diseases they're supposed to diagnose, what the clinical decisions might be, so on and so forth. And we've released a number of guidance on these, including IVD MIA, probably most famously, but others as well, use of software. And certainly these, how specifically to regulate these has, undergone constant discussion. But now we're really faced with the future of the so-called omics phase. Even though in some cases the future is here, we're really looking not just at next-gen sequencing, which may be the simplest case, which is a little scary when you think about all the problems we have with that, but also proteomics, epigenetics, the microbiome. People have talked about that in relationship to cancer. I mean, this is going to just get more complicated. And one of the features of all these is that, and I'll talk about this a little more later, often when you run these tests, you don't know exactly what it is you're going to end up diagnosing, which is why you're running these tests. This is certainly true in hereditary diseases, but it's also true in oncology. And that presents a certain, something new when you think about regulation. And so I'll talk about that to some degree. But because we're at the beginning of this, this is a discussion we will have with the community going forward. So I think, again, thank you, Adrian, for for covering all this. I just want to make one point here, though. So the definition of IVDs, what really is important when you think about this globally is intended use. So as was mentioned, who are you going to be testing? What is the analyte? What's the test? Are there sort of indications within that intended use? In some cases, what are the clinical decisions that will be made given those results? And we view everything in this light. So often it can be difficult for us to say, well, this is how we're going to treat NGS, or this is how we're going to treat proteomics, or this or that, because every case is different. And often what determines the path of regulation is what the intended use is, and sometimes based on the available evidence, that intended use will change during the course of development and review. So it is important when we think about all these examples to think specifically about what it is we're using all these tests for. And when you're developing, how am I going to define the intended use of this test? Now, as was mentioned before, we look at analytical and clinical performance, and so I won't belabor those points, but just to say this is pretty much a uniform standard we would hold to any test. What's going to change is the types of evidence that will be required to demonstrate those, depending on the type of test. The evidence for an NGS test is going to look a lot different than the evidence for a HER2 test, for instance. And so, there isn't going to be a one-size-fits-all approach, because there isn't a one-size-fits-all IVD. And this, actually, I realized recently, this can be very confusing to drug people, because that is a lot more of a well-patterned pathway. You do your pharmacology and safety, and at the IND stage, you have these things to demonstrate, and at the NDA stage, you have those things to demonstrate. Not that there aren't issues there, but IVDs are so different. I think this just really can present some confusion. Labeling is a big deal to us, and that's sort of one of the products of FDA review. So this evidence that we're talking about is going to end up in the label. And that's how we essentially get transparency to the performance of IVDs. And so when we are reviewing, we look not just at the analytical and clinical performance, but at the labels, and specifically what is said, the intended use, how the device is described, what the warnings and limitations are, all those things. And just to note, we do not actually look at clinical utility, although sometimes that's a byproduct of what we do. It's not our specific standard. So we don't, in some cases, our decisions could be taken as evidence of clinical utility, in some cases not. Now, I want to focus a little more on clinical validity, because we've talked a lot about evidence. There are sort of two approaches, general approaches people can take when submitting data to the FDA on clinical validity. One is clinical studies, which, as we've heard, can be difficult and expensive. Often you have to try to find samples somewhere, and good quality samples. You need to lock down the IVD. So usually there's an IVD being incorporated for companion diagnostics into a clinical trial. But there's also, we're going to see more and more of a big data approach going forward, where sponsors will rely on literature, they'll rely on case studies, they'll rely on databases, and we've already had experience with this to some degree. We have as long as the evidence is good, there's no reason why it has to be a clinical study. And I think as this conversation we started to have about databases going forward, I think this is going to be a very important part of FDA regulation in the future, and it will hopefully ease the pathway for everybody. So companion diagnostics, I think I don't really need to go over this, but just one thing to think about going forward is historically these have been one analyte, one test drugs, and obviously now with next generation sequencing, we're seeing the possibility of supplanting all those with a single test, and that's something we will be discussing going forward. So what is it about next generation sequencing that's challenging that's sort of new for the FDA? Besides all the other challenges that have been discussed. Well, the high information content certainly poses an issue. Just as was mentioned before, you're detecting giga somethings of data. And, certainly if we get to whole genome sequencing, and I don't know whether or not that'll happen on oncology, we're talking about over 3 billion bases in the human genome with millions of variants that any individual can return. So how are we going to analytically and clinically validate those? The other thing is often there's no really well predefined intended use in the same way that you might think about HER2. When you run NGS on somebody, and this is true in hereditary diseases, certainly, but also, will be in oncology if it's not already, you may have incidental findings, you may diagnose diseases or conditions other than the ones you originally started testing for. So if you start to think about some of the regulatory questions we have, well, now how do we apply our regulatory framework when because of the technology, you can't and you don't even want to define a very precise population? Of course, NGS tests are frequently modified, and those modifications, because it's still a new and rapidly evolving technology, and those modifications can affect performance. But we want to, at the same time, preserve the ability to do modifications because that's how innovation occurs. And you also have to accommodate specific testing needs. The pipeline for CNVs looks different than the pipeline for SNVs. I mean, there are a lot of similarities, but there are some crucial differences. And finally, right now, at least from the laboratory standpoint, there aren't that many systems out there. I mean, we've cleared 2 cystic fibrosis systems. There's nothing for oncology. There are a couple of cleared instruments. So right now, what laboratories are doing is they're essentially putting systems together in a mix and match sort of way, buying different components, customizing software. And so this leads to a lot of variability in the test. And so this will be a very important thing to consider going forward. And finally, just because of the nature of this, of the technology, we're going to be in many situations where we'll be detecting variants for which there won't be sufficient evidence, and there will never be sufficient evidence. They will be in rare populations. Some diseases are collections of rare variants. And so one of the big questions we have to ask is, how do we handle it? What is the threshold for evidence? And finally, and this was brought up before, what should they know? Like what sort of level of evidence is appropriate for patients and physicians? It may or may not be 100%. And how is that communicated in a way that's understandable? I mean, I think we would all acknowledge digesting this information in a way that people, particularly people with very little time or background in this, can understand is a real challenge. So what I wanted to do was talk briefly about, probably our best experience with this technology, which occurred in 2013 when we cleared the Illumina MiSeq platform, the MiSeq DX, so it's a single platform, as well as 2 assays on that platform for cystic fibrosis. And I think there are several lessons from this, or several approaches we took that we hope to build on in the future. So the first is that we ended up, the company separated the tool claim from the clinical claims. So what the MiSeq platform was cleared for was for sequencing, basically for doing panels out of blood for hereditary disorders, to paraphrase the intended use. It was not approved with a particular indication for a disease. That means, and this has been a very confusing point to people, you can't just buy the MiSeq and start running things and you're compliant with FDA. You need to develop a specific assay around that for hereditary diseases. And this is what Illumina did. So they developed 2 cystic fibrosis assays. One detected 139 variants that had good clinical evidence for a link to the disease in cystic fibrosis. They also developed a whole gene test that actually doesn't talk about clinical validity. It's for discovering any variant. It's for detecting any variant within the cystic fibrosis gene. So this is, I think there's a real advantage to this because now at least we have a cleared instrument that can be used for multiple purposes. And now if you're using that instrument within an intended use, but for another disease, you can basically use the MiSeq DX without having to further validate the instrument. The instrument is produced under GMP. So this is a big step forward, even though it doesn't get us all the way there. So how do we look at analytical and clinical validation, considering that the MiSeq DX could detect pretty much most variants in the genome? Well, basically the decision was to look at performance on a characteristic set of variants that represented different types of performance we thought the instrument needed to have. So GC-rich regions, different chromosomes, different proximities to the centromere, all sorts of things like that we looked at. And you can find all this. These clearances are posted on the FDA website. So those, the details, all the testing on the variants can be accessed. Same strategy towards the CF tests. The only thing here that needed to be clinically validated were these 139 variants that Illumina ended up getting clinical claims for. If you have this, we would diagnose you with cystic fibrosis. And we ended up using an external database, the CFTR2 database from Johns Hopkins as the clinical evidence. And I'll talk a little bit more about that in a bit because I think this is a good model moving forward. No, I'll talk about it now. The CFTR2 database had certain features that sort of allowed us to use it. It was curated by experts. It contained clinical data, and certainly we could just live on clinical data, but CF also had it has the benefit of having very good preclinical models. So you could take a variant and put it in a cell line and actually test whether that variant is affecting function. The cooperation of the patient community, patient organization for these efforts is going to be really important. We did have to ask them to version the database so we would know, for the purposes of capturing the information at a regulatory standpoint, where we were. And this just sort of lists some of the more detailed characteristics of CFTR2 database that I would point to as making it a very strong effort, including how it reviewed the literature, the degree of annotation, so on and so forth. But I wanted to sort of end this part leaving you with the sense of what a regulatory grade database could look like. So I think the question is, what's going to constitute a high quality database? How are we going to look at evidence coming out of it? And because we would like to consider being able to just look at databases and get clinical validity. It'll make everybody's life a lot easier. But we do need good annotation. We need good practices in the database, and the source of data has to be good. And so that's another conversation we are having going forward. This is something we're actually spending a lot of time thinking about. So I want to end talking again about evidence, but from a different standpoint. So this is sort of a little, not a pyramid, but a little scale I made that, obviously in the end, what we're looking for is good evidence in one form or another, to push forward patient care. And right now in oncology, this comes in the form of targeted therapies, which are supported by trials or other research evidence. These need high quality data. And so I would argue that at the base of this, we have to have, not just for a clinical practice, but even to generate the evidence, we're going to have good quality IVDs that we understand. And so I just want to bring up one point that because we see a lot of clinical trials for companion diagnostics, and one of the areas of concern is about the quality of data that's coming out, and in particular with the sort of environment we have now with LDTs and FDA-approved things and different technologies for, say, detecting BRAF. And a lot of trials will use, and sometimes they have no choice, to use local testing results in a patient's EHR or medical record. When these results, and we can't assure that these results are comparable, and they may not, in some cases, they may in other cases affect the quality of clinical trials. So we do have to ask, when we're seeing, 60% response rate in a clinical trial, I think it would be good to ask what degree of that is variability in responses underlied by the test. And so this is just one example of how, no matter what the solutions are, it is important to think about where this data is coming from. Often by the time it gets into the medical record, it is a result. BRAF positive, BRAF negative. You don't know anything else about it. Just in the interest of time, I'm going to skip over this. There are a lot of ways to address this, when designing a clinical trial, but at the very least we have to be aware of these issues. Now, we're going to be dealing with a lot of variations of NGS sequencing in the future, liquid biopsies, cancer panels, moving to whole exome, whole genome. We are open to considering a variety of approaches, and but at the core, all these, no matter what these technologies are, it's going to come back to the analytical and clinical validation. And I just wanted to mention a couple of things about the LDTs. I don't need to show the previous slide, but obviously, as we've mentioned several times, there's now this current proposal, and at this point, it's just a proposal for public discussion out that would first collect basic information on LDTs through a notification process, use a public process to obtain input on risk and priority for regulation, and then phase in the regulatory framework over a very long period. So with the highest risk things coming in first and the lowest risk things, coming in and being cleared in 9 years. When we've had some carve-outs for various things like tests for unmet needs and so on and so forth. The details are all in the guidance. There was also a webinar recently, which was publicly posted. So I think that'll go into the nitty-gritty of the details. That's a little dry. But I did want to end by talking about interacting with the FDA, because I think this is really the path for success for individual developers and for the field. So for people who are applying to FDA or are thinking about developing, we do have specific pathways. Pre-submission, there's no rule about when you have to meet with them. You can meet very early on, even during a conceptual phase. We have a lot of interactive review during a submission. And I think that's very important. This is not you send it in and then you hear a no. There's a lot of discussion that goes on during a review. We have a lot of resources posted online, including guidance and some basic resources. Specifically on LDT policy, and I really want to point it out, this is intended to be a public discussion period right now. So in addition to the webinar we had, there's a public docket, so please comment. We want you to road test this and let us know what you think. We will analyze this. We're also going to have a public workshop in January. So that is, there will be lots of opportunities for interaction about that. And we do want input. And finally, we have a lot of discussions. Obviously, we come out and talk at in various settings, including this one. We participate in a lot of internal and external efforts, workshops, other opportunities. We want to be interactive. We want to hear what's going on in the community, and we want to have our regulation promote, obviously our mission is to protect patient safety while promoting public health, but we also want to preserve innovation, because that's really, that's extremely important to bringing things to patients. So sorry I went over time, but thank you.
Last but not least, the last David.